Everyone, today I'll be going through the International A-Level Biology content for edexcel for Unit 1. This will include all the chapters. I will link this PowerPoint down below in the description box. Feel free to use it if the quality isn't high enough or you want to go over anything. Carbohydrates, lipids, and proteins. Basically, what you need to know is the structure of water and what it enables it to do. So water has a dipole nature because the hydrogen atoms in the water are more positive than the oxygen atoms. So they are attracted to each other as the opposite charges attract. So ions are able to dissolve in the water because of this dipole nature. So for example, ion like chloride is negative. It will be attracted to the hydrogen atom and form bonds with it, so it dissolves in the water. And hydrolysis is a reaction where water is used to break down molecules like carbohydrates, lipids, and proteins. Carbohydrates are composed of carbon, hydrogen, and oxygen. And you need to know that monosaccharides are the soluble carbohydrate monomers made of a single sugar unit. They are soluble and they are the simplest sugar unit. Two monosaccharides can join in a condensation reaction to form a disaccharide joined by glycosidic bonds. So a carbohydrate monomer joins by glycosidic bonds. A maltose is a disaccharide that's composed of two glucose. Sucrose is composed of a glucose monosaccharide and a fructose. Lactose is composed of glucose and galactose. And polysaccharides are several monosaccharides joined together by glycosidic bonds. For example, glycogen in animals as an energy store and starch in plants. Lipids are nonpolar molecules that are insoluble in water. Basically, polar molecules have OH groups that are polar and can form hydrogen bonds with water, so they are able to dissolve in water. But nonpolar molecules do not, so they cannot dissolve in water and they are hydrophobic. Lipids are used as long-term energy reserves in organisms. Lipids can either be saturated with no carbon-carbon double bonds or unsaturated with carbon-carbon double bonds. So, basically, there are two types of lipids. Saturated, which actually cause cardiovascular disease, and unsaturated, which are basically the healthy kind of lipids. We'll go over the differences between them later. For example, triglycerides. It's an example of a lipid. It's made of 3 fatty acids and 1 glycerol joined by ester bonds. Fatty acids in the glycerol and the triglyceride or overall can differ in chain length, presence, and number of double bonds. So a triglyceride can either be a saturated one or an unsaturated one, just like any fatty acid can be. Proteins are made of amino acid monomers joined by peptide bonds. The primary structure is the amino acid sequence, and the secondary structure is the folding, using hydrogen bonds. The tertiary structure is the 3D folding by ionic bonds, etc. The quaternary involves more than one polypeptide chain getting bonded together. Here are some questions involving water. They are all from past paper questions. So first off, explain how water is involved in the transport of molecules in living organisms, because water is a polar solvent that dissolves ions to transport them. And why is glucose more soluble in water than, for example, any other ion? Because glucose has more OH groups, so it can form more hydrogen bonds with water. And why are fatty acids insoluble? Because they have nonpolar tails and cannot form hydrogen bonds with water. Explain how the properties of water molecules result in surface tension. This is a 3-mark question. Water molecules are dipolar, first mark, therefore they can form hydrogen bonds with each other, resulting in cohesion between them, which results in an inward force at the surface of the water. Water has a high specific heat capacity as there are many hydrogen bonds that would need a lot of energy to increase the temperature of the water. This ensures aquatic life as temperature is constant for enzymatic activity. this is the answer to the question that would go anything from why it takes a lot of energy for water molecules to be broken down, like a large body of water, why it doesn't evaporate, or why the temperature is mostly constant in water bodies. This is very important because if it wasn't, all the aquatic animals inside the water would die. This right here shows the dipolar nature of water. You are often asked to draw it in past paper questions. Here are some questions for carbohydrates. A very common one is describe the structure of starch. Starch is a polysaccharide and it's made of two different polysaccharides. It's made of amylose and amylopectin, and amylose and amylopectin each have a different structure, but they do have stuff in common. You can be asked to compare between amylose and amylopectin. Basically, they are both A-glucose, like alpha glucose monomers, and they are both joined by glycosidic bonds. They both have 1,4 glycosidic bonds. A 1,4 glycosidic bonds means that the first carbon is joined to the fourth carbon on the second alpha glucose. But the difference is amylopectin has 1,6 glycosidic bonds. So it, other than having 1,4 glycosidic bonds, it also has alpha glucose monomers that are joined the first carbohydrate to the sixth carbohydrate. Okay, so if you are asked to describe the structure of starch, you will say that it is compact, it is insoluble, so it doesn't have any osmotic effect. If it was soluble, the osmotic balance in the organism's body would be disrupted. Amylose is also linear, it is coiled if you are asked to compare between them. Okay, you can be asked why amylopectin and glycogen are good energy stores. Okay, so they are both branched, so they are easy hydrolyzed into glucose for energy. And they are compact stores, so you can store a lot of energy in a tiny bit of space, and it is insoluble, so it has no osmotic effect. And since they are polysaccharides, they are energy stores, since polysaccharides are stores of glucose, or like the monomer is glucose. Basically here it shows the difference between alpha glucose and beta glucose. In the alpha glucose, the hydrogen is above the OH group, hydroxyl group, and in the B glucose, the hydrogen is below the OH group. Okay, and you are also required to know what each disaccharide is composed of. So maltose is composed of 2 glucose, 2 alpha glucose. Sucrose is composed of 1 alpha glucose and 1 fructose. Lactose is composed of 1 alpha glucose and 1 galactose. Here we have questions about lipids, and this is a very important diagram. You're often asked to show how a triglyceride is formed. So you have 1 glycerol and 3 fatty acids, and they turn into a triglyceride. This reaction is a condensation reaction. You release 3 water molecules. Here we have a few common questions about lipids, and this is a very important reaction. It's how triglycerides are formed. It's important that you don't forget to draw the water molecule, as there is a mark for it. These are questions about proteins. This is the most common question about proteins, how the primary structure determines the 3D shape of the protein and its properties. Here we have 1b, it's about circulation, blood vessels, heart, and atherosclerosis. This, two very important questions. Why do mammals need the circulatory system, like unlike insects, for example, and advantages of double circulation? Please memorize these before your final exam. This is the blood clotting cascade. So first, when a blood vessel is damaged, the platelets release thromboplastin, and with the presence of calcium ions. Thromboplastin converts prothrombin into thrombin, and thrombin catalyzes the reaction of turning fibrinogen into fibrin, which is insoluble. Fibrin forms a network of fibers that trap platelets and blood cells into an insoluble mesh. This is the Bohr curve. It's important to do many questions about this. The concept isn't quite hard. You have to explain the shift of the curve, the Bohr shift. And here, these are very important. You are often asked to draw the structure of the artery. You have to label the lumen inside and the layer of endothelial cells. You have to label the tunica media, the one in the middle. It has muscles and elastic fibers, and the layer on the outside, tunica externa, that has collagen fibers. Veins and capillaries aren't asked about as often, but remember that capillaries are 1 cell thick and they are made of one layer of endothelial cells. It's very important to remember how arteries are adapted to their function. The last 3 points are specific to the aorta artery. Here we have the structure of the heart. You have to remember what the names of each veins are. The most commonly asked one is the aorta and the vena cava. You have to also know the names of the valves. Here we have the cardiac cycle. The first step is the arteriosystole, ventricular diastole, when the atria contracts to push blood into the ventricles, and then ventricular systole, when the ventricles contract to push blood out of the heart. And here we have cardiac diastole. All chambers are relaxed and blood flows into the heart. Atherosclerosis is the hardening of arteries caused by a build of plaque or atheroma. So how does this happen? First, the endothelial lining in arteries is damaged due to high blood pressure or cholesterol or smoking, and that leads to an inflammatory response causing white blood cells to move into the artery. Cholesterol, calcium ions, and blood cells build up on the artery wall into an atheroma, and then the atheroma hardens into plaque. And then that leads to the further narrowing of the artery, restricting blood flow, increasing the blood pressure, damaging endothelial lining all over again. This is very dangerous if it happens in the coronary artery, as it reduces blood supply to the heart muscle, so deprives the heart muscle from oxygen and nutrients needed for aerobic respiration. So the muscle, goes for anaerobic respiration, which produces lactic acid. The pH gets too low, the enzymes are denatured, and the heart muscles die. So no blood is pumped around the body, and the person can get a heart attack and die. 1C. You have to know the definition for correlation and causation and the definition of risk. You have to know why each risk factor is a risk factor. So why antioxidants reduce the risk. They reduce the risk of cardiovascular disease because they reduce free radicals that damage endothelial lining that would otherwise cause an atheroma. And why age is a risk factor, why gender is a risk factor, and why diet is a risk factor, why smoking is a risk factor. Why do people underestimate the risk of cardiovascular disease? For example, high salt diet, or high cholesterol diet, high energy diet, or smoking on cardiovascular disease. Because cardiovascular disease takes so long to develop, it has long-term effects and takes a long time to develop. People will think they're fine and won't notice an actual difference in their body until it's too late. And they see other people around them doing the same things and think they are fine, so they just assume they are going to be fine too. They mostly ask this question for 3 marks, so you only need to memorize 3 of those points and leave the rest. Okay, if you want if you want to plan an experiment on the effect of a risk factor on CVD like vitamin C, you have to get a large sample of healthy volunteers. Why does it have to be a large sample? For reliability and repeatability. Why do the individuals have to be healthy? For validity. Because if the individuals already have cardiovascular disease or at high risk of cardiovascular disease, then it's too late to see if this factor will increase the risk or not. The individuals have to be of similar age, gender, mass, lifestyle, and diet. You divide those individuals into two groups. One is given, for example, a vitamin C supplement or a certain type of antioxidant, and then the other group is given a placebo. Why is a placebo given? So that we know that the vitamin C is what is causing the change, not the mental factor of it. And then you monitor the incidence of heart disease over the group over a long period of time. This study might be unreliable. Why? Because not all factors can be controlled. We can't have all the volunteers of the exact same mass, of the exact same lifestyle, of the exact same diet. And then you might have an unreliable estimate. There is no clear definition of a high salt diet, and like if you have a group of smokers, how many cigarettes are smoked by each person differ. The treatments of cardiovascular disease and the risk of each one. Antihypertensives reduce blood pressure, but they cause drowsiness, heart palpitations, swelling of the feet and ankles, a persistent cough. Statins reduce levels of LDL cholesterol because it is a risk factor to have a high LDL to HDL ratio. So we lower the LDL to HDL ratio using statins, but it can cause things like diabetes or liver problems. Anticoagulants it reduces the formation of new blood clots, so it reduces atherosclerosis, especially in the coronary artery. Can cause excessive bleeding if an injury occurs. Platelet inhibitors work in a similar way. They also reduce the formation of new blood clots, so the risk of blood vessel blockage is reduced, but can also cause excessive bleeding in case of an injury. An experiment you would have to describe is to compare the vitamin C content of 3 fruits. So you have to collect fruit extracts from all 3 fruits, get beakers with a certain volume of DCPIP, titrate the drops of fruit extract 1, the first fruit, till the color changes from blue to colorless of the DCPIP, record the drops needed, repeat for the other fruits on new DCPIP in a beaker of the same volume. Repeat the whole experiment 3 times for all fruits. Use calibration curve to get the concentration, and control the storage fry and age of fruit. Why the risk decreased over years? So for example, it gets you 2 graphs, one from the 90s and a new current one. Why is, for the same risk factors, why is the risk reduced for people? Because better healthcare, there are more pills developed, new statins, all of those, and people are more aware of risks and lifestyle. Okay, the difference between reliability and validity. Reliability is when you can repeat the experiment multiple times and get similar results, like here. Validity is when the results are correct. 2A, we've got cell membranes and the transport across cell membranes. So first, the cell membrane controls the movement of substances into and out of cells. It has proteins and glycoproteins embedded into the membrane, has surface receptors to bind to things like hormones. The structure of the cell membrane is a phospholipid bilayer membrane. It has phosphoid heads outside as they are hydrophilic and interact with polar aqueous environment, while the fatty acid tails are nonpolar or hydrophobic, so they orientate themselves away from the aqueous environment. The fluid mosaic model is a model of the cell membrane. It's fluid due to its fluidity of phospholipid bilayer. It allows molecules to flow within it, and the phospholipids are constantly moving. It's mosaic because of the transport proteins, receptor proteins that have various shapes and sizes embedded randomly into the cell membrane. Cholesterol is found in the membrane to give it stability and reduce the fluidity. The nonpolar part of the fatty acid tails is embedded into the fatty acids, and the OH group is with the phosphate heads to keep it in place. Here are the types of transport: active, which uses energy from ATP; passive, that doesn't use energy. Simple diffusion is for small non-polar molecules like CO2 and O2. And facilitated diffusion is for polar ions that need a polar protein channel to go through it. Osmosis is for water. Endo and exocytosis, bulk transport for large molecules. And active transport is for molecules against their concentration gradient. You have to know the adaptations of gas exchange surfaces. They have a thin alveoli wall that reduces diffusion distance. They have many alveoli that increase the surface area for diffusion, a capillary network to maintain concentration gradient, and good ventilation for concentration gradient. Explain the factors affecting the cell membrane permeability. Temperature, because as temperature increases, the rate of diffusion increases. Permeability increases because the kinetic energy of molecules in the membrane increases, so they move more, the phospholipids move more. Cholesterol reduces the movement of phospholipids as it combines with fatty acids, holding the fatty acid chains together in place, so phospholipids move less. It's vital to say phospholipids move less. I don't know why I didn't write that. Write that. Okay, fatty acid chains are nonpolar and allow nonpolar substances to diffuse into cell. Polar ions need a polar channel to get into the cell through the cell membrane. 2b talks about enzymes. You have to know how to define an enzyme. Enzymes are biological catalysts that increase the rate of reaction by lowering their activation energy of the reactions they catalyze, either intracellular reactions, interactions in cells. Mustafa, how many times would I record the thing? And then how nucleotides join. Let's imagine this. Oh, let's turn on the pen. Here, those are the pentose sugars. Here is the phosphate group. So to have the sugar backbone, the two nucleotides join by phosphodiester bonds. It's called phosphodiester because what's joining them is the phosphate group. And then this just moves on to have a polynucleotide chain. This is the sugar backbone. And then we have hydrogen bonds between the bases. So here we have those bases. And then on the other side, we'll have another polypeptide chain. Here I'm showing you the structure. All the images are from Save My Exams. I hope they don't copyright me. But anyways, I forgot I put a diagram already. I didn't have to draw it for you guys. And adenine and thymine, they have 2 hydrogen bonds. Cytosine and guanine, they have 3 hydrogen bonds. And you can be asked to compare the structure of DNA and RNA. DNA is double-stranded. It has thymine, and DNA has deoxyribose sugar. While RNA, it's single-stranded, has uracil instead of thymine, and it's a ribose sugar. They both have pentose sugar. Ribose and deoxyribose are both pentose sugar. They have a phosphate group. Both have phosphodiester bonds between nucleotides, and they both have the adenine and guanine bases. Here it's also showing you the structure. The purines have a single carbon ring. The pyrimidines have a double carbon ring structure. Now we have DNA replication. DNA replication is described as semi-conservative. Semi-conservative because one DNA molecule will divide into 2 DNA molecules. Each DNA molecule has a newly synthesized strand and an old paternal strand from the old DNA. It's shown here. Here we've got 2 strands. One is purple, one is blue, and then one of the old blue strands is here on the left, and then the new strand is on the right. It's commonly asked to describe semi-conservative replication or just DNA replication, and it's always semi-conservative. So you have to write the steps. The double helix unwinds as DNA helicase breaks the hydrogen bonds. One strand is used as a template strand, and complementary base pairing occurs, attaching free nucleotides to template strand. DNA polymerase moves along the strand, joining adjacent nucleotides by phosphodiester bonds in condensation reaction to make the new DNA. How do we know that it's semi-conservative? How did we prove that? Okay, so two scientists called Messelson and Stahl, they proved it by growing bacteria in N15. It's I forgot the name. Oh right, isotope. Isotope is the word. Okay, N15 is an isotope of nitrogen. So it's a heavy nitrogen isotope. Oh, it's right there. I could have continued reading. Okay, and then they transferred it to nitrogen 14 culture, which is the normal light nitrogen, to find out each DNA molecule has one strand with N15 and one strand with N14. So here we're showing the experiment, because they looked at each one, and each one has one N15, one N14. It means that when it was replicating, it used one old strand and generated one new strand. Nature of genetic code. The order of bases in DNA make up genetic code. Introns are the coding parts of DNA, and exons are the non-coding part. So genetic code is a triplet code, so 3 nucleotide bases make up a codon which codes for a particular amino acid. So 3 bases code for 1 amino acid. So the first feature of the genetic code is that it's a triplet code. And then it's a non-overlapping code, so each code is only read once, and then as the amino acid is recruited, and then the ribosome moves on to a new codon, and so on. It's a degenerate code, so different codons can code for the same amino acid. For example, the codons CUU and CUC both code for the amino acid leucine. This means that some mutations will have no effect on the organism, since the same protein will still be produced. So, since the genetic code is degenerate, that's a very huge advantage for living organisms, since silent mutations can mutate. Silent mutations can be undergone. Silent mutations can happen. Silent mutations can happen with no effect whatsoever. And it's a universal code. This isn't often in the mark scheme, but it's still a point in the syllabus, so we have to say it. Universal code, so all organisms use the same genetic code. Bacteria, bonobos, and bananas all contain DNA made up of the 4 nitrogenous bases that are found in humans. Here, it shows you the transcription part of protein synthesis. Transcription is when, it's the part where the DNA in the nucleus is turned into mRNA. Here is the translation part of protein synthesis, which happens at the ribosome. So that mRNA code that has already been transcribed is translated into a protein or polypeptide. You have to memorize the steps of transcription. So first off, the RNA polymerase enzyme attaches to the promoter region of the DNA and then unzips the gene. RNA polymerase moves along the gene and unwinds the gene by breaking the hydrogen bonds between the nitrogenous bases. The free ribonucleotides in the nucleus bind with complementary base pairs on the antisense strand of the gene. The A binds with U and C with the G. The free nucleotides are joined by condensation reaction to form phosphodiester bonds. This is guys, I'm kind of worried actually. The new mRNA formed peels away from the coding strand, and the DNA rewinds, which happens when the stop codon is reached. Translation: mRNA moves from the nucleus into the cytoplasm and attaches to the ribosome. tRNA carries an amino acid and attaches to the... Just memorize this. there's no much explaining to do. If you don't get it, you can watch another YouTube video. So, because I'm just summarizing, I'm not doing much. I think my brother is sick. I don't know though. 2C. The first lesson is about mutation. Mutation is a random change in DNA sequence coding for a polypeptide. A point mutation is a mutation that involves a change in the DNA base sequence at a single location. So everything we have in the syllabus is a mutation that involves a change in the base sequence at a single point. But some mutations take place in multiple places in the body or at the autosome cells. So every single cell has that mutation in the whole body. So for example, substitution, insertion, and deletion. Insertion is when an extra base is inserted into DNA sequence, often results in a non-functional protein, as all codons after insertion are affected. This is called frame shift, as the code is non-overlapping. Deletion is when a base is removed from the sequence, resulting also in frame shift and a likely non-functional protein, as all codons after deletion are changed. Wait, do I have a picture for this? No. I'll just draw for you guys. So let's say we have A, U, C, G, A, T, something. And then you insert. Well, you know how those are triplet codes. Each 3 code for 1. If you add a little, let's think, what will we add? We'll add another A here. Okay? So now we will probably have a non-functional protein because let's say this codes for like leucine protein, amino acid I mean. And this codes for, I don't have amino acid names memorized. We'll call it Melissa. Okay, now, after the mutation, what happened is that this isn't the triplet code anymore that's going to be read, because those 3, AGA, is going to be the new amino acid. So it's going to translate into new amino acid. And then after this, let's say TAT. Instead of it being GAT, and then the one after ATG, we have a new amino acid. And each one after that will also have a new amino acid. So none of the amino acids after the frame shift will be correct. And same with deletion. Substitution mutations include silent mutations. Substitution is when one single base is substituted by another, replaced by another. Silent mutations, the mutation does not alter the amino acid sequence of the polypeptide. This is due to the degenerate code of the nature of the genetic code. So as we said, genetic code is degenerate, which means that multiple codons code for the same amino acid. So basically there will be no difference because somehow by chance the mutation caused a degenerate okay, moving on. Missense mutations. The mutation alters a single amino acid in the polypeptide chain. For example, sickle cell anemia is caused by a single substitution mutation changing a single amino acid in the hemoglobin protein. Amino, is it amino or is it amino? Amino, right? Have I been saying amino acid the whole video? Amino acid. Okay, so missense means a whole new amino acid will be produced. Nonsense is when the base is substituted, replaced by a base which codes for a stop codon. A stop codon is the one that tells the mRNA to stop translating. So the polypeptide or protein will be shorter, and yeah, anything after that will not be coded for, since there are no complementary tRNA molecules with anticodons. So basically the mRNA is coding at the ribosome, and then the stop codon comes, and there is no tRNA with an amino acid for that. So it just doesn't continue. Inheritance. Every cell contains 2 copies of each gene, 1 from each parent. We have 23 pairs of chromosomes, and the chromosome in each pair are called homologous chromosomes. In each pair, 1 comes from the mother and 1 from the father. You have to know these definitions very well. They are often asked about, very, very often. I don't know why I didn't write genes, but you were also often asked about what a gene means. Gene is a part of DNA base sequence coding for a polypeptide. Genotypes is a combination of genes, although combination of alleles in the individual. Phenotypes is the characteristics expressed in an individual caused by a mix of genes and environmental factors. Alleles are different forms of a gene. A dominant allele is always expressed when it is present. A recessive allele is only expressed when it is in homozygous form. So both alleles are recessive. So you don't have any other allele to express. So the recessive allele is expressed. Cystic fibrosis. Cystic fibrosis, you have to memorize it very well. It's often asked about too. So what causes cystic fibrosis? Cystic fibrosis is caused by a mutation that causes a faulty CFTR transport protein in the surface membranes of epithelial cells. The SFTR is a chloride ion transport channel. So the chloride ions do not exit the cell. So the chloride ions stay in the cell. So water does not leave cell by osmosis, since the chloride ion concentration in the cell is much higher than outside, since no channel protein is helping the chloride ions exit the cell. So then the mucus on the epithelial cells is not diluted by the water. This results in thick, sticky mucus. It lines the tubes and ducts in the gas exchange tract, the digestive tract, and reproductive system. The sticky mucus increases the chances of lung infection and makes gas exchange less efficient. Microorganisms become trapped in the sticky mucus, causing illness. Cilia cannot move the mucus because it is too sticky. So the cilia cannot beat to move the mucus away, so it's just stuck in the airway. It reduces ventilation. It reduces gas exchange since the airway is so inflamed. Low levels of oxygen in the mucus, it is harmful bacteria can live in these conditions. Gases such as oxygen cross the walls of the alveoli into the blood system by diffusion. The sticky mucus makes it harder for diffusion to take place. For the digestive system, the problems caused by cystic fibrosis is that the pancreatic duct becomes blocked by that sticky mucus, impairing the release of digestive enzymes. The lower concentration of enzymes within the small intestine reduces the rate of digestion. Because of this, the food is not fully digested and not all nutrients can be absorbed. The pancreatic enzymes can also become trapped behind the mucus, blocking the pancreatic duct. The enzymes damage the pancreas, damage to the cell walls in the pancreas that produce insulin. Insulin is involved in the control of blood sugar levels. A form of diabetes can be the result. So the main problem is that since pancreatic duct becomes blocked, there are no enzymes in the small intestine to break down the large insoluble food molecules into small soluble ones that can be absorbed, so it is not absorbed, so that leads to malnutrition. In the reproductive system, we are mostly asked about females with cystic fibrosis. They have a reduced chance of becoming pregnant because a mucus plug develops in the cervix, and this stops sperms from reaching the egg. So because the mucus is so sticky, the sperm is stuck in that mucus before reaching the cervix or the fallopian tube, so the egg cannot be fertilized. Genetic testing. The DNA is tested to see whether it contains the known base sequence for the most common mutations that cause the genetic disease. So, genetic testing can be used in multiple ways. It can be used to identify adult carriers. They can choose to adopt or not have kids. This is the answer to the question which asks you why, because of the genetic testing, less kids now have a genetic disease, for example, cystic fibrosis. Genetic testing can be used for testing embryos. Amniocentesis involves inserting a needle into the amniotic fluid to collect cells that have fallen of the placenta and fetus, and then it is tested for the mutations. Cryonic villus sampling, a small sample of placental tissue, is removed either through the wall of the abdomen or through the vagina. Testing before implantation for IVF. It is possible to test an embryo before it has been implanted into the uterus. A cell can be removed from an embryo. The cell can then be analyzed and used to decide whether to place the embryo into the womb or not. Problems with genetic testing: if it's so convenient and you can eliminate people with such hard genetic diseases, why do we not all get genetically tested before we implant kids? Because of ethics. There are religions that are against abortions, as life is a sacred gift from God. The baby has a right to live. They view that discarding IVF embryos is like murder. Sick babies are less worthy of life, and this causes social stigma around illness. If we abort every baby with the genetic illness, people will think that they are less worthy of living than normal human beings. This may cause a miscarriage of fetus, and you can get false positive results, and that would lead to a loss of a healthy baby. Oh, we are done.